Hello again, Bio 220 students, and welcome to this lesson in Chapter 10, Classification of Microorganisms. Now, as far as classification, there is one field which involves taxonomy. It is the science of classifying organisms based off of degrees of similarity among those organisms. And then there is systemics or phylogeny, which involves the study of the evolutionary history of organisms and how different species have branched from previous ancestors. As far as a timeline of developing this science, in 1795, Linnaeus developed the thought of having kingdoms for plants and animals. And in the 1800s, bacteria and fungi were put into the kingdom Plantae. In the kingdom Protista was also proposed for bacteria, protozoa, algae, and fungi. In 1937, the prokaryote was introduced to distinguish cells that did not have a nucleus. And in 1968, Murray developed the kingdom of Prokaryotes, and in 1969, Whittaker developed the five-kingdom system. As far as the level above kingdom, or the three domains, it was developed by Wallace in 1978 based on sequences of nucleotides in rRNA, so looking at genetics and not just physical similarities. What was developed is the three kingdoms, or the three domains, which are eukarya, which includes all eukaryotic organisms. These can be animals, plants, and fungi. And there are also the other two domains, which are bacteria and archaea, which, despite being prokaryotes, they are so different to each other that they warrant separate domains. And this is just showing similarities, or characteristics, I should say, of the three domains used today. Now for the three domains. Eukaryotes, they originated from infoldings of prokaryotic plasma membranes, and through endosymbiosis, bacteria developed into organelles. For example, it's proposed that chloroplasts and mitochondria came from the engulfment of smaller bacteria by larger organisms, microbes. And this is the model of the origin of eukaryotes, which involves some early cell that branched off into bacteria, and then eventually that branched off into bacteria, and then into another branch that would eventually divide into archaea and eukarya. Eukarya eventually developed foldings where their genetic code surrounded was enveloped, and eventually that eukaryotic cell, in some cases, eventually went on to engulf smaller bacteria that would eventually become mitochondria and chloroplasts. Reasoning behind this proposed model is because chloroplasts and mitochondria do have DNA that is unique from that is in the nucleus. Now, a phylogenetic tree is a grouping of organisms according to common properties, and these can involve fossils or, more modernly, genomes, or the genetic code of that entire species. Groups of organisms have evolved from a common ancestor, and each species retains some characteristics of its preceding ancestor. For scientific naming or scientific nomenclature, common names vary from language and geography. This is a sort of way of how one particular species will be called one thing in one area of the world, but will be called a very different term in a different area of the world. But to help have some unified way of collectively naming organisms, the binomial nomenclature was used, which involves two specific terms, the genus as well as the specific epithet, or the species. And this collection of a genus and species is used as its binomial name, which is unique for every organism. For a taxonomic hierarchy is a series of subdivisions developed by Linnaeus to help distinguish plants and animals. There is a eukaryotic species, which is a group of closely related organisms that can breed between themselves. Essentially, organisms or eukaryotic organisms in the same species can breed from different members of that species. For example, a species of dog where different types of dogs like golden retrievers and labradors are able to successfully breed with one another. And this is just a way of showing the different levels of organization and hierarchy, starting with all organisms and then breaking off into domains, which include eukarya, archaea, and bacteria, eventually breaking off into a kingdom, which for eukarya could be fungi, animalia, or plantae, and then getting into phylum, class, order, family, and then genus, and then species. The combination of genus and species is the term used, or is what are used for the binomial name unique for any particular organism. Now, classification of prokaryotes. Well, prokaryotic species involve a population of cells with similar characteristics. A culture will be bacteria grown in a laboratory setting. A clone is a population of cells derived from a single parent cell, and a strain is a genetically different cells within a clone. For classification of eukaryotes, there are protista, which is essentially a catch-all kingdom for a wide variety of organisms that can be autotrophic or heterotrophic, and they'll be grouped into clades, which are groupings that are based on their ribosomal RNA. There is another eukaryotic kingdom, which is fungi, which are the chemoheterotrophic organisms that can be unicellular or multicellular. Their cell walls are made out of chitin, and they can develop from spores or hyphal fragments, parts of the original organism. There is the kingdom Plantae, which are multicellular organisms that have cell walls made out of cellulose and will utilize photosynthesis. And then there's the last kingdom of Animalia, which are multicellular, they have no cell walls, and they are chemoheterotrophic. As far as classifying viruses, there is not any one particular domain for viruses. They range incredibly heavily, and they do not use a baseline of a cell unit. They instead actually require a host cell for reproduction. But in order to group them, there are viral species, which are populations of viruses with similar characteristics, for example, similar genes or similar protein coats that occupies a particular ecological niche. Classifying and identifying organisms. Classification is just simply placing organisms into meaningful groups of related species, and it will involve listing the characteristics of known organisms in that grouping. There is identification, which is the matching of characteristics of an unknown organism to a list of known organisms, which is beneficial in a clinical lab setting. In the case of bacteria, there is Berge's Manual of Determinative Bacteriology, which provides helpful identification schemes for identifying bacteria and archaea. And then there's the Approved List of Bacterial Names, which lists species of known classification. In clinical microbiology, lab requisition forms are used to note types of specimens collected and tests to be conducted. There are transport media, which is used to collect and transport pathogens to a laboratory. And this is just a sample clinical microbiology report. Form. Further classification can involve morphological characteristics. It's useful for identifying eukaryotes based off of the traits that can be observed physically. However, it will not be too helpful about the actual phylogenetic relationships, where phylogeny involves the actual genetic sequence of an organism and not just its physical, observable characteristics. There is differential staining, where gram staining, acid-fast staining, but they're not useful for bacteria without cell walls. In the case of biochemical tests, they can determine the presence of bacterial enzymes, which can be used to classify or identify bacteria based off of what enzymes they will typically produce. And this is just a way of going through a logical sequence of steps to determine what bacteria or microbe you are working with. For biochemical tests, there is the rapid identification method, which performs several biochemical tests at the same time to look for different properties or different biomarkers or different molecules while running at the same time. Results of each test will be assigned a certain number to help organize the variety of information. First, serology. The species that studies, or the science that studies serum and immune responses in that serum. There are microorganisms that are antigenic. They stimulate the body to form antibodies in the serum. In an anti-serum, a solution of antibodies is tested against an unknown bacterium. In the slide agglutination test, bacteria agglutinate when mixed with particular antibodies produced in response to the bacteria. In serological testing, that can be used to differentiate between species and strains within a similar species. For serology, there are enzyme-linked immunosorbent assays, ELISA assays, where known antibodies and an unknown type of bacteria are added to a well, and a reaction will identify or help identify that particular bacteria. There's also Western blotting, commonly used in pharmaceutical labs, which identifies antibodies in a patient's serum, and it can be used to actually confirm human immunodeficiency virus infection, or HIV. For phage typing, it's a test for determining which phages a bacterium is susceptible to. Bacterial phages are things that will attack bacteria. Typically, they will be viruses. On a plate, clearings called plaques appear where phages infect and lyse or rip apart bacterial cells. Fatty acid profiles. FAME, which is fatty acid methyl esters, will provide profiles that are constant for a particular species. There is also flow cytometry, which uses differences in electrical conductivity between species, or it also involves using fluorescence to tell species apart. Then there's DNA-based composition. It's essentially a measurement of the amount of guanine and cytosine in a species' DNA. Two organisms that are closely related will typically have similar proportions of guanine and cytosine. Which are nucleic acid bases in DNA strands. DNA fingerprinting, which is essentially electrophoresis of restriction enzyme digests of an organism's DNA. Restriction enzymes will essentially cut DNA in specific regions that have a specific lettering code. And then those could be separated by length in a process of electrophoresis, which will help spread a collection of DNA strands and spread them in a gel based off of length. There's also it's used for comparing fragments from different organisms and provides information on genetic similarities of differences. The key here is that restriction enzymes, no matter what DNA is mixed with those enzymes, whether from a mouse, a human, or different microbes, as long as there's a certain restriction enzyme will only cut or slice a strand of DNA at a very unique nucleic acid base sequence. That's how we're able to differentiate strands of DNA. And this is just an example where electrophoresis, or a difference of voltage, was used to move different lengths of DNA strands from different samples across a gel. And then the different bands that are shown are just different groupings of DNA strands. Now there is amplification techniques to help take a little bit of DNA and increase its concentration significantly. This typically involves the use of polymerase chain reaction, PCR, to amplify or grow the amount of DNA of an unknown microorganism that cannot be easily cultured. Nucleic acid hybridization will measure the ability of DNA strands from one organism to hybridize with DNA strands from another organism. Hybridization is simply how we have two strands of DNA, or a double helix, in our genetic code. That's because one strand will hybridize or link up with another strand. So in this situation, one strand from an organism is mixed with strands of DNA from another organism to see how well they pair up with each other. When there's a greater degree of hybridization, there's typically a greater degree of relatedness between those two organisms. And this is just a diagram showing what was just discussed. Another type of blotting, which is southern blotting, uses nucleic acid hybridization to identify unknown microorganisms using known DNA probes and then it looks to see whether DNA from that unknown organism will hybridize with what is already known. There are DNA chips, which consists of a microarray, or contains a large amount of DNA probes with a certain sequence of DNA. And it's useful for detecting pathogens by the hybridization between the known probe and the DNA from the sample specimen. And then fluorescence will be used to determine the concentration of hybridization, or how much DNA strands have been suitably hybridized. These DNA chips are also used in ancestry tests to determine similarity between a person's genetic code to populations from, say, parts of Europe, Asia, Africa, or indigenous populations throughout the world. Now, DNA chips can involve ribotyping, which is sequencing the particular type of ribonucleic or ribosomal ribonucleic acid. There's also fluorescent in situ hybridization, FISH, where fluorescent DNA or RNA probes will be used to stain microorganisms that are being targeted. What this does is it helps determine the identity, the abundance, and relative activity of microorganisms in a particular environment. When putting classification methods together, there are dichotomous keys, or identification keys based on successive questions. These can be typically yes or no questions. Does this particular specimen have this trait or this trait? Yes or no? And then go on further down the key. There are also cladograms, which are maps that show evolutionary relationships among organisms based on their genetic rRNA sequences. And then this is just a way of showing how cladograms are built not simply by physical characteristics, but by similarity in genetic material. And that concludes this lesson in Chapter 10. In Chapter 10 on microbial classification. As always, it is a pleasure to have each and every one of you in this online iteration of microbiology, and catch you all in the next learning module. Peace.